A rat had basically no brain, but it could still see, hear, smell, and feel. How could that be possible? Life without a brain, neuroradiological and behavioral evidence of neuroplasticity necessary to sustain brain function in the face of severe hydrocephalus. A 2-year-old rat R222 survived a lifetime of extreme hydrocephaly affecting the size and organization of its brain. Much of the cortex was severely thinned and replaced by cerebrospinal fluid, yet R222 had normal motor function, could hear, see, smell, and respond to tactile stimulation. The hippocampus was malformed and compressed into the lower hindbrain together with the hypothalamus, midbrain, and pons, yet R222 showed normal spatial memory as compared to age-matched controls. Bold MRI was used to study the reorganization of R222's brain function, showing global activation to visual, olfactory, and tactile stimulation, particularly in the brainstem cerebellum. The results are discussed in the context of neuroadaptation in the face of severe hydrocephaly and subsequent tissue loss, with an emphasis on what is the bare minimum for survival. This rat named R22 did have a brain, but its brain, affected by a condition called hydrocephalus, had compressed and collapsed as it filled with fluid, and many of the functions that would ordinarily be carried out in the brain had relocalized to areas that weren't taken over by fluids. This provided the tools for Ferris, who was a psychology professor at Northeastern, to investigate how powerful the brain remains, even when tight on space. This, he says, might even influence the ever-present goal of machine learning. How small can you be and still get the job done? Pretty small, it turns out, at least in R2's case, but this efficient use of space is dependent on the brain's capacity to reorganize. This ability, known as neuroplasticity, is a widely documented phenomenon, but such an extreme example was rare, says Ferris. In R2's case, the processing of visual input was distributed over much of the remaining brain, and the same thing with smell and touch. What at first the scans suggested to be a brainless rat was actually a rat with a brain that had been pushed out of the way and flattened like a pancake and kept working. Ferris's lab, the Center for Translational Neuroimaging, originally received R2 as a part of a group of test animals from Alexion Pharmaceuticals for a study on aging, and Ferris kicked off the study as it would any similar project by taking preliminary scans of the animals. While doing so, Ferris's colleague flagged out something on the computer by saying, "Look at the screen. The rat doesn't have any brain." In Ferris's words, R2 was one of the nature's miracles, since living for 2 years, like living for more than 70 human years, is unlikely with such a severe deformity. Given that, they had this unique opportunity to try to understand how this animal survived. Successful adaptation to this extreme abnormality suggested the animal had it since birth. First, though, the team had to measure just how similar R2 actually was to the other rats in the cohort. To assess memory, the researchers placed each animal in a plexiglass box to observe how it responded to a new space and the objects within it. They then observed the rats navigating a maze in a further display of memory as well as of spatial learning skills, and measured physical behavior as the animals walked a balance beam. In all but one of these tasks, R2 performed just like the other rats. The exception was exploring a novel environment. The other rats moved around and showed interest in their surroundings, and R2 stayed put. On its own, though the latter behavior wouldn't indicate something debilitating, says Paris. This failure to explore is common among anxious animals, so beyond the possibility that it experienced heightened anxiety, R2 functioned normally and importantly appeared normal on sight. And yet, brain areas as seemingly crucial as the hippocampus, which processes memory, were so distorted that Ferris and his team couldn't even identify them on sight. It was only after they traced chemicals in the brain that they were able to verify that indeed the hippocampus was that squished, displaced object pushed towards the back of the brain. The lower part of the brainstem had everything collapsed in it. The animal just defaulted to what evolution gave it in the beginning, along with all the other animals, to help it survive. For rats, that's enough. Most of their life is spent working through their nose. But could humans, too, get by with less brain power? Thank you for watching the video. If you like the video, don't forget to like and follow my page so that you can get other updates in the future. Thank you.